Hello students, we are going to talk about potentiometry titration. Let us see the principle. When a metal strip is placed in a solution of its own ions, there are two possibilities or tendencies. First one is that metal atoms may dissolve in the solution as positive ions, leaving electron on the electrode. Second case, the metal ions may take up electrons from the electrode and get deposited as neutral atoms. Okay, तो इस केस में क्या हो रहा है, वन इलेक्ट्रोड एक पे मेटल्स आयंस मेटल एटम्स जो है वो सॉल्यूशन में डिसॉल्व होंगे इन एज पॉजिटिव आयंस और वो इलेक्ट्रॉन्स लीव करेंगे ऑन द इलेक्ट्रोड. ये इलेक्ट्रॉन्स जो है वो मूव करेंगे एंड मेटल आयंस दूसरा जो इलेक्ट्रोड रहेगा उसमें की जो मेटल आयंस है वो इलेक्ट्रॉन्स एक्सेप्ट करेंगे और डिपॉजिट करेंगे एज न्यूट्रल एटम्स. ओके, इस तरह से पोटेंशियल डिफरेंस जो है वो सेटअप होता है बिटवीन इलेक्ट्रोड एंड द सॉल्यूशन. ओके, सो पोटेंशियोमीटर ये जो इंस्ट्रूमेंट है दिस इज यूज्ड टू डिटरमाइन द पोटेंशियल डिफरेंस बिटवीन द रेफरेंस इलेक्ट्रोड एंड द indicator electrode, and these two electrodes form electrochemical cells which are dipped into the solution to be analyzed. So measured potential can be used to determine the quantity of analyte in terms of concentration. So जो solution का हमको concentration find करना है इस तरह से हम वो potential use करके उसका concentration find कर सकते हैं. Okay, so this one example which I have discussed now. So one atom is converted into its ions and it is giving out electrons. So zinc is giving out electrons. Okay, and the other electrode, that is the cathode. Cathode pe Ag, silver ions, they are accepting these electrons and deposited as a neutral atom. Okay, so these two solutions, they are connected by salt bridge. Okay, and this is connected by, so ये जो potential difference is created, okay, ये हम measure करेंगे with the help of potentiometer using, you can say, galvanometer. So electrode potential जो होता है that is tendency of the electrode to lose or gain electrons. तो standard potential जो होगा that is potential of the pure metal atom, pure metal when it is dipped in the 1 molar solution of its own ions at 25 degree, और उसको हम कहेंगे standard electrode potential. Then you have two types. One is oxidation potential. The potential of the substance to get oxidized is called as oxidation potential, and reduction potential is the potential of the substance to get reduced. Now we will talk about the instrumentation part. Potentiometric cell has following parts, that is electrodes, salt bridge, analytical solution, and galvanometer. Okay, so in this example, we can see copper electrode is dipped into the copper sulfate solution. Zinc electrode is dipped into the zinc sulfate solution. These two are connected by salt bridge, and here you can see the galvanometer which will read the potential difference between these two electrodes. So working, we will see now the potentiometer consists of two electrodes whose reduction potential differ inserted in a test solution. Okay, जो voltmeter attach किया है वो potential difference measure करेगा. One of the electrodes is reference electrode और जो दूसरा electrode है वो है आपका टेस्ट इलेक्ट्रोड. ओके, तो टेस्ट इलेक्ट्रोड हमेशा क्या रहेगा, मेटल immersed in a solution of its own ions whose concentration we want to find out. Okay, or it can be a carbon rod electrode in a solution which contains ions of interest in two different oxidation states. Now oxidation takes place at anode. For example, इस केस में हम देखेंगे zinc. Okay, it is giving out electrons, it is losing electrons. Okay, so zinc is getting oxidized into zinc ions. Reduction is taking place at cathode. तो cathode पे क्या हो रहा है, copper ions are taking up electrons, copper is getting reduced. Okay, so इस तरह से जब ये redox reaction होता है, that time the potential is developed and we are going to measure that potential difference by using galvanometer. Okay, now electrode potential जो है that is the potential of the electrochemical cell, और उसको हम ये फॉर्मूला से एक्सप्रेस करते हैं, E of cell is equal to E of indicator, that is potential of the indicator electrode minus E of reference, that is potential of the reference electrode. Now plus E of J, this is the potential which is developed across the liquid junction at each end of the salt bridge, but it is very less, so we are going to so it is negligible. So our final equation will become E of cell, that is potential of the cell will be the potential of indicator minus the potential difference of the reference electrode. Okay, so here we can see again zinc, which is a 1 molar solution. Zinc and anode is dipped into the solution of 1 molar zinc sulfate, and copper is electrode is dipped into the 1 molar copper sulfate solution. Here zinc is losing electrons, it is getting this is oxidation. Copper is accepting electrons, so this is a reduction, and migration of ions, which is taking place through the salt bridge. Okay, and this circuit, the flow of electron will take place from anode to cathode. Okay, and whatever is potential difference which is generated, we are going to measure with the help of voltmeter. Now we will discuss the reference electrodes and different types of indicator electrodes. We will discuss standard hydrogen electrode, Caloral electrode, silver chloride electrode, which are the reference electrodes. And in the indicator electrode, we will discuss metal electrode, hydrogen electrode, twin hydrogen electrode, and glass membrane electrode. So let us first discuss reference electrodes. तो सबसे जो कॉमन है and it is mostly used widely, that is standard hydrogen electrode. So this is standard hydrogen electrode potential is defined as the potential which is developed between hydrogen on platinum and hydrogen ions of the solution when hydrogen gas at pressure of 760 mm of Hg is in equilibrium with the hydrogen ions of unit concentration. Okay, so here we have a platinum foil which is coated with platinum black, and it is dipped in the 1 molar HCl solution. Pure hydrogen gas will be bubbled continuously at 1 atmospheric pressure. Platinum will act as a conductor, inert, and it facilitates in attaining the equilibrium. तो इसका जो electrode potential होता है वो है 0.00 volts. Okay, so hydrogen gas, that is gas electrons, and this is converted. This is a reaction of the cell reaction will be represented like this: H2 gas converted into 2H plus ions and 2 electrons. So, okay, so this is the diagram of hydrogen electrode. Okay, you can see here hydrogen gas is bubbled at 1 atmospheric pressure, and this is your platinum wire electrode, platinum electrode, which is coated with the platinum black. Okay, and this is dipped in the solution of 1 molar HCl solution. So finally divided platinum coated on the platinum foil, you can see here. So when your hydrogen electrode is coupled with the proper type of half cell, then your hydrogen electrode can reverse its behavior and it can work either as cathode or anode. Okay, when it behaves as anode, in that case the hydrogen will be oxidized to hydrogen ions, and when it is acting as a cathode, the reverse reaction will take place. Okay, now there are some advantages and there are some limitations to standard hydrogen the electrode. That is, it cannot be used in a solution containing strong oxidizing agents, and second, it is difficult and expensive to maintain, and excess of hydrogen bubbling out will carry little HCl with it, so the concentration of H plus ion decreases. In that case, it is difficult to maintain the concentration of HCl at 1 molar. Then we will discuss the second one, that is Caloral electrode. This is most commonly used electrode in the potentiometry. There are 2 glass tubes, one outer one and inner one, and the outer tube contains solution of potassium chloride, and inner tube contains a slurry of mercury and mercurous chloride. Okay, there is a small pinhole. Okay, there is a small opening which is connecting these two both, and with the saturated solution of KCl. Okay, platinum wire is placed inside the slurry. Okay, so the ceramic filter will act as a salt bridge. Now the reaction, Caloral electrode reactions can be expressed like this: 2 Hg plus 2 chloride ions giving Hg2 Cl2 and 2 electrons. तो इसका जो Caloral electrode का potential है, that is found out to be 0.2444 at 0.244 at 25 degrees Celsius. Okay, we can see here. Okay, the 2 glass tubes you can see, the inner one and the outer one. Okay, outer tube, we can see the solution of KCl. HG, there is a mercurous chloride and HCl solution. Here the opening is there. Okay, so and this is a solution of KCl. So the potential of this electrode will change with the chloride ion concentration. And generally, जो potassium chloride use करते हैं, that is employed in 0.1 molar, 1 molar, and saturated solution. So the saturated Caloral electrode is the most commonly used, and it is widely used as a reference electrode. The advantages: concentration of chloride ions do not change even when some of the solvent get evaporated. Okay. Secondly, it generates small junction potential, so it is more accurate. But there is a limitation that mostly saturated solution of KCl is used, and that is temperature dependent. Then we will discuss the next one, that is silver chloride electrode. Now this is also similar to Caloral system. This is most widely used in a reference electrode, and this consists of a silver wire coated with silver chloride. The coating may be electroplating, and your coated wire is placed in 1 molar solution of silver chloride. So the cell reaction can be represented as Ag plus Cl minus gives AgCl and 1 electron. तो इसका जो potential है वो है 0.199 volts. But this is advantages. It is easy to handle and cost effective, but sometimes it shows reactivity. Okay, so this is an example. This is a diagram of silver chloride electrode. Okay, we can see the silver wire which is immersed in the solution of potassium chloride, and that is saturated with silver chloride. There is a porous junction we can see here. Now we will discuss the indicator electrodes. Now in the indicator electrodes, the first we will discuss is a metal indicator electrodes. The metal electrode develops electric potential as a result of redox reaction at its surface. Now there is a first order electrode jahan par that involves metal in contact with its own ions. For example, silver ions, zinc in presence of zinc ions. Okay, so what is happening here? Silver ions taking up electrons and deposited as silver. So the potential is 0.8. Okay, now limitation is metallic indicator electrodes are not very selective and respond not only to their own solutions but also to other more easily reduced cations. Many metal electrodes can be used only in the neutral or basic solutions because they dissolve in the presence of acids. Now, second order electrode is the one that responds to the presence of precipitating or complexing ions. For example, silver wire could serve as an indicator electrode for chloride. So Ag plus Cl ions will give AgCl plus electrons. The potential is 0.199. Okay. Then the third one is inert electrode. The most commonly used indicator electrode is known as inert. And these electrodes are not involved in the half-cell reaction of the electrochemical species. Typical inert electrode is platinum, gold, carbon. Inert electrodes are responsive to any reversible redox system, and these are widely used in potentiometric work. Now we have glass membrane electrode. Now this glass membrane electrode is most commonly used indicator electrode, and that involves ion exchange reaction. The membrane is made up of chemically bonded Na2O, silica, Al2O3. So we have a glass bulb which is fitted with solution of HCl and KCl. Silver acetate coated with silver chloride is inserted as electrode. Okay, so here we can see this. There is a glass bulb with a long neck, and it is a special type of cell which is selectively permeable to hydrogen ions. Okay, we have the inside this glass bulb, we have the reference solution of hydrochloric acid. Okay, that is usually 0.01 normal, and we have a silver wire which is coated with a thin layer of silver chloride that dips into the electrolyte solution, and it serves as an internal reference electrode. Okay, in the potentiometry, glass electrode is often coupled with the saturated Caloral electrode. And here you can see this So inside the glass bulb, we can see the solution, which is HCl solution. You have the silver wire coated with silver chloride, and you have a thin glass membrane. So this is magnified, and how it is, what is the structure is shown here. So there is actually SiO2 gel layer on the glass surface. Okay, so 0.1 mm glass membrane with exchangeable sodium ions. And the potential of this cell is E is equal to K plus 0.0591. Okay, so working, first glass will absorb water, then the hydrogen ions can move in the direction of lesser concentration. They will replace Na plus ions and the other ones in the glass membrane. Okay, so SiO2 plus Na and H plus will replace this. So SiOH plus ions and Na. As a result of diffusion and exchange process, the potential develops on each side of the glass membrane. Now, the potential is not affected by presence of strong reducing oxidizing agent. It can operate over wide pH range. It responds fast and function well in the physiological system. This is used in pH meter. Okay, so this is how the pH meter, the glass electrode looks. Then we'll discuss hydrogen electrode. Hydrogen electrode is used in the acid-base titration, but it has very limited application because many organic substances directly react with hydrogen gas. Then we have quinhydrone electrode that contains a solution of quinone and hydroquinone prepared from quinhydrone. Okay, so 2QH giving Q plus H2Q. This is simple and easily attains equilibrium. This is employed in the presence of mild oxidizing and reducing agent at pH of 8. This is sensitive to high concentration of salt and oxidizing and reducing agent. Okay, so the electrode consists of, there is an inert metal electrode, that is a platinum wire in contact with the quinhydrone crystals and a water-based solution. Quinhydrone is slightly soluble in water, dissolving to form a mixture of two substances, quinone and hydroquinone, with the two substances present at equal concentration. Each one of the two substances can be easily oxidized or reduced to other. Potential at the inert electrode depends on the ratio of activity of two substances, that is quinone and hydroquinone, and also the hydrogen ion concentration. The electrode half reaction will be hydroquinone giving quinone plus 2H plus and 2 electrons. For practical pH measurement, a second pH independent reference electrode such as silver chloride electrode is also used. This reference electrode does not respond to pH. The difference between the potential of two electrodes depend on the activity of H plus ions in the solution. So it is this potential difference that is measured and it is converted to pH value. Now this quinhydrone electrode is not reliable above pH of 8. Okay, this is also unreliable in the presence of strong oxidizing and reducing agent, which would disturb the equilibrium between quinhydrone and quinone. It is also subjected to errors in solution containing proteins or high concentration of salt. Now, other electrodes commonly used for measuring pH is a glass electrode, hydrogen electrode, antimony oxide electrode, and the ion sensitive field effect transistor electrode, that is ISFP. Now, the salt bridge part, this is a U-shaped tube filled with the inert electrolyte. Glass tube bridge is there that consists of gel plus Ki or Na2 sodium sulfate. There is a filter tube bridge which is containing filter paper, KCl or NaCl. Now the function, it will allow the electrical contact between the two solutions, prevent mixing of two solutions and maintain electrical neutrality. Okay, so this we have already discussed. Now titration part, quantitative measuring procedure in which the liquid solution is added to the mixture until some distinctive feature, signal or endpoint is observed. So potentiometric titration is a volumetric method in which the potential difference between the two electrodes is measured as a function of added reagent volume. Okay, so this is our solution, that is an analyte. We'll be adding the reagent from the burette. This is a titrant. The two electrodes are dipped into this and it will measure the potential difference between the two electrodes. Okay. So we will see what is the application of potentiometric titration, that is precipitation titration, complexation, titration, neutralization titration, and redox titration, that is oxidation reduction titration. So first we will see what is precipitation titration. Volumetric method based upon the formation of slightly soluble precipitate are called as precipitation titration. So we have already studied what is precipitation titration. So what is the precipitating agent? Silver nitrate. Okay, that is why it is called as argentometry titration. Now the precipitation titration is very important because it's a perfect method to determine halogens and some of the metal ions. And then complexometry titration. Complex ions are produced from the reaction of metal ion, which are the electron acceptors with the electron pair donors. The donor species, we call it as ligands. They must have at least one pair of unshared electrons for the bond formation. In the neutralization titration, there is a standard solution of strong acid or bases. When a single solution is sufficient for the titration of given type of analyte, it is convenient to have standard solution of both acid and base available in case back titration is needed to locate the endpoint more accurately. And then we have the oxidation reduction titration. The redox type of titration is a titration based on the redox reaction between the analyte and the titrant. The redox reaction may involve use of the redox indicator and the potentiometer. An example of redox titration is treating the solution of iodine with the reducing agent and starch as the indicator. Iodine forms a intensely blue colored complex with starch. Iodine is reduced to iodide by thiosulfate. And when iodine has spent, the blue color disappears. This is a iodometric titration. Applications of potentiometry. You can carry out analysis of pollutants in water. You can do drug analysis in the pharmaceutical industry, in food industry for analysis of quality, biochemical and biological assay or the analysis to check the quality of cosmetics, and also used as analytical tool in textile, paper and paints, explosive energy and more. In agriculture, nitrate, ammonium, chloride, potassium, calcium, iron, cyanide in salt, plant material, fertilizers and feedstuff, detergent manufacturer, calcium, barium, fluoride for studying the effects on the water quality, food processing, nitrate, nitrite, in meat preservatives, salt content of fish, meat, dairy products, fluorine in drinking water and other drinks, calcium in dairy products and beer, potassium in fruit juices, and corrosive effect of nitrates in the canned foods. Okay, so this is all about potentiometry titration. Thank you.